Hello guys, welcome to IB360. So today we will start with a new chapter which is cell metabolism, and the first topic under cell metabolism is enzymes. So enzymes are proteinaceous substances. Okay, they are commonly proteinaceous substances, and they are well known biochemical catalysts. So they are biocatalysts which catalyze thousands of chemical reactions inside our body, and that is why they are very famously known as biocatalysts. You know what are catalysts? Catalysts, they have the capacity to accelerate a reaction. That means increases the speed of the reaction, make the reaction very smooth without themselves undergoing any change. So enzymes are acting as catalysts inside our body. Each cell has over 5,000 different enzymes, and these enzymes, they are catalyzing thousands and thousands of various metabolic reactions inside our cells, body cells. So they are usually globular proteins. Okay, proteinaceous substances, they are usually globular proteins. In my protein class, remember I was talking about the classification of proteins, and as per shape is concerned, proteins can be globular or fibrous. Enzymes are globular. All enzymes are globular proteins, and this idea that all enzymes are globular proteins, all enzymes are globular proteins, with some exceptions, with the exception of recently discovered RNA enzymes, RNAs, ribozyme, which is known as, except that all enzymes are globular proteins, because then that is not a protein, at least not a protein only. So how can it be a globular protein? But this sentence that all enzymes are globular protein, this was proposed by Sumner, biologist Sumner, in 1926. Okay, that all enzymes are globular proteins, and the term enzyme was coined by Kuhn in 1878. But interestingly, their functions or their significance was not yet known. But in much later, like almost 20 years later, in 1897, it was Buchner who discovered enzymes. So Kuhn is known to coin the term, and Buchner is known to actually discover enzyme. So Buchner was also actually awarded Nobel Prize in the same year for this discovery. Actually, Kuhn in 1878, he coined the term enzyme for catalytically active substances which were previously called ferments. Okay, which were previously called ferments. So from the term ferments, the term enzyme came into existence, and that was done by Kuhn. But in 1897, what happened? Fermentation of sugar was very common in those days. Actually, started right from those age only. So, with accidental discovery, Buchner found that fermentation of sugar is not only caused by living yeast cells, but also yeast extract. That means the juices which we can get out of yeast. So the extract obviously possessed biocatalysts which were required for the process, and from there the term enzyme got its significance. Here N means, this part actually means in, and zyme, this part means yeast, in yeast. because the extract which Buchner found could actually catalyze the process of fermentation of sugar. Okay, sugar fermentation by yeast extract. So he noticed that the juices or the items or the item which basically catalyzed this process of sugar fermentation came out of yeast. So from then only the term enzyme got its significance, that is in yeast, inside yeast. Whatever was found inside yeast, that was termed as enzyme. But now we know that all enzymes are obviously not found inside yeast. There are many enzymes which are found inside human cells, over 5,000 different enzymes to catalyze different reactions. Later, you will also know that enzymes are highly specific. That means a particular kind of biochemical reaction can be catalyzed only by a particular enzyme. Only by a particular enzyme under particular conditions of temperature, pH. pH means level of acidity or basicity. So this is a very basic introduction for enzymes. Enzymes are mostly proteins. They are biocatalysts, and the term enzyme was coined by Kuhn, but enzyme was discovered by Buchner. And inside our body cells, there are over 5,000 different enzymes which catalyze biochemical reactions. See, every biochemical reaction is catalyzed by a specific enzyme. So here the specific enzyme part is really very important. Let us move on to the next topic, which is exoenzyme and endoenzyme. So enzymes are mainly functional inside living cells, that we know. But as found out by Buchner, they can be extracted from the cells and made to catalyze reactions outside the living cell. What did Buchner do? Buchner, from yeast cell, he took out the enzyme and made it work extracellularly. That means outside cell in sugar fermentation. That was the story. So from there we get an idea that enzymes can operate outside the cell also. So from there these two terms came into play. One was exoenzyme, one was endoenzyme. So in nature, some enzymes are secreted by living cells and made to perform extracellular catalysis. Examples would be digestive enzymes. So exoenzymes, digestive enzymes. Also lysozyme in tears. It's an antibacterial enzyme, lysozyme. So all these are examples of exoenzyme. All enzymes are secreted or synthesized within the cell, but they would be exoenzyme or endoenzyme, depends on their job or their application, that where are they performing their job. Are they inside the house or the outside the house? If they are performing the activity outside the house, which is the cell, they would be exoenzyme. Okay, so several categories of enzymes, several enzymes of medical and chemical importance, they belong to exoenzyme. Like for example, rennet tablets from renin of calf stomach, which is used for coagulating milk protein casein during preparation of cheese and other milk products, is also an exoenzyme. Why? Because they are functional outside the living cell. They are functional. They're synthesized within the living cell, obviously, but their function, see this arrow suggests that the enzyme is moving out of the cell. Okay, so let me do this in some other color. So functional outside the cell. Then comes endoenzyme. Endoenzyme is functional within the cell. Endoenzymes can be enzymes of Krebs cycle. You know what is Krebs cycle? Krebs cycle is a phase which includes a chain of metabolic reactions during cellular oxidation, during cellular respiration. We know at individual levels the cells are respiring themselves. So Krebs cycle is a chain of processes which involves respiration of the cell inside the mitochondria, and also enzymes of glycolysis we can right, even enzymes of glycolysis. Glycolysis is again a part of cellular oxidation, but this takes place in cytoplasm, and Krebs cycle takes place in mitochondria. So all these enzymes are operating, or they're functional within the cell. So they're endoenzyme. So this is the overall idea of exoenzymes and endoenzymes. Okay, in fact, the enzyme which Buchner discovered was also an exoenzyme because it performed outside the yeast cell. It was functional outside the cell. Okay, let us move on to the next part, which is some new words you will learn today: substrate, reactant, and product. So, the biochemical which is acted upon by an enzyme is known as substrate. So, what is substrate? The biochemical acted upon by the enzyme. Okay, that is a substrate. And in case biochemicals are involved in a reaction, the same are called reactants. So reactants would be more than one substrate. So this is the enzyme. This one is the enzyme. Let me. So this one is the enzyme. Okay. And in the first stage, you see substrate is entering active site of enzyme. This is the substrate which is entering the active site of enzyme. This active site is important because enzymes, they have got specific spots where the substrate can fit, because you see the substrate has a specific shape. So if the substrate comes from this angle, or if the substrate comes from this direction, it won't fit the enzyme. Only if it comes from this direction it will fit, just like a jigsaw puzzle. So that is why the active site, we must know the positioning or the location of the active site for fitting in the substrate. The next thing is formation of the enzyme substrate complex. This is the second stage. The enzyme substrate complex is formed, which is when the enzyme and the substrate exactly fits each other, like the individual elements in jigsaw puzzle. The third step is the enzyme product complex. So see, as the substrate has been attached, fixed properly with the enzyme, the substrate undergoes changes and we get the product. So then the substrate undergoes changes and we get the product. The enzyme doesn't undergo any change. It remains exactly the same, but it is the substrate which is undergoing some change under the influence of the enzyme, and we get the product. But now, as the product has been formed, it will no longer be in need of the enzyme because already the reaction has taken place and we have got the resultant product. So now it will be time for the product, the fourth stage, to leave the active site. So leaving active site of enzyme, these are the products, which are obviously different from the substrate, as you can see. So ultimately, if we want to write them in an equation, it will be sort of like this. We have the substrate and under the influence of the enzyme we get the product. Or in a better way we can write something like this, that we have the substrate plus enzyme, which gives us the substrate enzyme complex, AC complex, substrate enzyme complex. So this was an individual entity, this was an individual entity, but together they form a singular entity, which is the second step here. From substrate enzyme complex we get product enzyme complex, which is the third step. And from product enzyme complex, product is released and enzyme stays as it was. So this is the flow chart for substrate to product pathway basically. And if there are more than one substrates, they would be called reactants. Okay, if there are more than one substrates, they would be called reactants. Fine. Then biological importance of enzymes. See, enzymes, they are biocatalysts, we had already heard. There are thousands of chemical reactions occurring inside our body always, and all are mediated by enzymes. They operate at biological temperature, for human 37 degrees Celsius, which is room temperature inside our body, and the body must maintain this particular temperature. So that's why we are not poikilothermos. We have a mechanism to regulate body temperature because enzymes are involved. Enzymes, they're very fussy. They're very fussy about temperatures, about acidity, and they also require a specific pH for that matter. And that is why the pH, there is different pH operating in different parts of the body for the enzyme specificity. Okay, and as they operate under favorable conditions, they force the organism to live in a favorable habitat. The fifth point is they are highly regulated. Enzymes are a result of translation of genes. We know our DNA, which is inside the nucleus, stores information, and some of the information is also meant for enzymes, that what kind of enzymes the cell will synthesize. So there are several other factors which are influencing this process of enzyme synthesis from DNA. And if the cell wants, or if the cell feels like it doesn't require that enzyme much, then it can switch off its mechanism of synthesizing that particular enzyme. Then that will be repression of genes. It can ask the gene to shut up. But if it sees, or if it feels that it really needs that enzyme to perform its activities, its processes, then it will activate the genes. That would be activation, and the genes will continue synthesizing those certain enzymes. So then the enzymes can be functional or non-functional in the cells. Okay, so this is all about today. This is the basic introduction for enzymes. We will talk about enzymes again in the next class. Till then, stay safe. Thank you guys.